This is the work of my recently graduated student, Yuheng Wang. So we all know that environments vary, they vary over time, they vary across space, and evolutionary biologists have long been interested in how environmental heterogeneity shapes genetic and phenotypic variation. And to study these kinds of questions, a number of years ago in my lab, we established a set of experimental fly populations that are maintained with and without these different forms of environmental heterogeneity. And we've used these populations to study a number of questions, including how environmental heterogeneity affects levels of inbreeding depression, sequence variation, additive genetic variation, and adaptability. But today I want to talk about a recent project using these populations to ask questions about expression, evolution, and plasticity. So these flies were first collected from the field about a decade ago. That's supposed to be 2005. And from that, we maintained in a large lab population on standard cornmeal food, and we refer to that as our grand ancestor. We also created another large lab population maintained on a cadmium-enriched diet. We call that the ancestral cadmium. And another large lab population maintained on a salt-enriched diet called the ancestral salt population. Those ancestors were crossed, and from that cross, we created 20 smaller replicate populations that were divided amongst 4 selective treatments. One where they lived just on cadmium, one where they lived just on salt, one where there was temporal variation alternating each generation between the two, and one where there was spatial variation, half the population living on one food type, the other half on the other, and then mixed each generation. After 120 generations of evolution on these food types, we collected RNA-seq data from young larvae that lived for 12 hours on either cadmium or salt food. And so the first question that we're going to look at is sort of harkens back to ideas from Waddington and West Eberhard and many others, and it's this one. Do the evolved differences between populations that are adapted to just cadmium or just salt, do those differences reinforce or oppose the plastic responses of the naive ancestor? So to look at this, we'll consider data from our grand ancestor and from these cadmium and salt adapted lines. And we screen for genes that meet 2 criteria: genes that have a large plastic response in the ancestor across these 2 diets, and also that have an evolutionary divergence between the cadmium and salt lines. There's 97 genes that meet our criteria for this. And again, the question is whether the evolved difference between the cadmium and salt selected lines will be in the same or opposite direction to the plastic response of the grand ancestor to these 2 diets. And here's sort of a simple version of looking at the data. This is the plastic response of the diet ancestor, and here's the evolved divergence between the cadmium and salt selected lines. And this plot you can see from here that most of the genes fall in these 2 quadrants, indicating that the evolved response is in the opposite direction from the plastic response. So 87 of them, the 97 genes, show a counter-gradient response. You can also, this is analyzed at the gene level. You can do it at the replicate population level, which is arguably better. It shows the same thing. And this is several other recent expression studies that have found this kind of counter-gradient response. It seems to be a bit of an emerging pattern in expression data. And at least in our case, where we looked at it, we looked in ours in a little bit more detail, and I don't have time to go into it, but there seems to be a very simple explanation for that. That's sort of cartooned here. So for a number of these genes, there's a sort of a low or moderate level of expression in the salt diet for all 3 population types. But for populations that don't have evolutionary history in the cadmium diet, those genes show a perturbed response, but the population that's adapted to life in cadmium shows more or less a normal level of expression, and that's what gives rise to this counter-gradient response. For the remainder of the talk, I want to focus on the evolution of plasticity expression itself. So when a population is first exposed to a novel diet or a novel food, it can have a variety of transcriptional responses, and those plastic responses, some of them might be good, some of them might be bad, and many of them will probably be neutral. And the lack of plasticity at other genes can be good, bad, or neutral. And it's really only in populations that evolve with environmental heterogeneity where selection will have that opportunity to adaptively shape reaction norms. For example, increasing plasticity when it would be good to have different levels of expression in different environments. So our sort of very basic question is, are patterns of plasticity more adaptive in our heterogeneous selection regimes than in our homogeneous selection regimes? And our secondary question is whether the adaptive evolution in plasticity occurs equally with temporal variation versus spatial variation. But as I said, the genes can respond in different ways, and so our first challenge is what are a set of genes where we'd expect selection to increase plasticity or to decrease plasticity? And I'll talk first about increases in plasticity. So the way we do this is by making use of the diet-specific ancestors that founded all of the treatment populations. So to look for genes where we expect an increase in plasticity, we essentially look for genes like the one that I've cartooned here. So here I'm showing you expression for the ancestral cadmium and the ancestral salt population in the cadmium diet and in the salt diet. And these 2 points here represent the different adaptive states, right? So ancestral cadmium in cadmium and ancestral salt in salt. And so we expect plasticity here because it seems like it's good to do different things in the two environments. That is, the adaptive states across environments are very different, and there's actually very little plasticity in each ancestral population. So we identify a set of 109 genes this way. And remember, these are genes that are identified using these diet-specific ancestors. And now we're going to look at the level of plasticity in these genes in the treatment populations that were founded from those ancestors. So that's what's shown here. This is the average level of adaptive plasticity across the set of 109 genes for these 5 replicate populations in each treatment. And positive values here mean that plasticity is in the adaptive direction. And what you can see is that there's low levels of adaptive plasticity for the regimes with homogeneous selection, and higher levels of plasticity in the regimes with heterogeneous selection, significantly so in spatial, and at least there's a trend that is more so in spatial than in temporal. So those are genes that we expect selection to increase plasticity. We also identify a set of genes where we expect selection to reduce plasticity. But here the data are the patterns not really as we expected. So we expect a reduction in plasticity in the heterogeneous regimes. That's not what we see in general. It seems somewhat, maybe a little bit lower in spatial than these regimes, but the temporal regime actually has higher levels of plasticity, significantly higher than spatial. So from this, it would seem like things in temporal are not very well adapted based on this pattern of plasticity. But in fact, it turns out that's misleading. And the reason it's misleading is that plasticity tells us about change across environments. It does not tell us how good expression is in either environment. For that, what we'd really like to know is how close is the level of expression to the optimal level of expression. Now, of course, we can't really know the optimum, but we can take as a sort of first approximation of that the level of expression of the diet-specific ancestor, so an ancestral called cadmium in the cadmium diet, and take that as the optimal level of expression, and take the non-adapted ancestor as sort of a bad level of expression, and create this sort of arbitrary scale from 0 to 2. And then if we measure some other expression in other populations, we'd say that this orange population has a closer distance to the optimum than the blue population. So we can do that for all these genes in these gene sets. And so here's the average adaptive distance from the optimum for that gene set where we expected increases in plasticity. So remember, low values here are good. And what we see is that both the heterogeneous regimes have a better level of expression than the non-adapted ancestor. That's a significant difference in the cadmium diet, and the same trend is apparent in the other diet. Okay, so that's for the genes where we expected adaptive increases in plasticity. We can do the same thing for genes where we expected adaptive decreases in plasticity. And remember, the pattern with respect to plasticity here didn't look like there was much adaptation going on. But in fact, when we look at it using this measure, it looks like in these heterogeneous regimes, they're doing very well, thank you very much. They're doing better than these non-adapted, homogeneously selected populations. That's true in cadmium, and that's actually true in salt as well. And also it looks like, remember, for temporal, the temporal regime, it seemed particularly bad, and they're actually doing very well. At least looks like it's better than spatial. we did another screen to look for a different set of genes that where we expected a reduction in plasticity, and that set is shown here. So here's how much plasticity we expected, and we again find the same pattern where it looks like there's more plasticity in the temporal regime than in the spatial regime. So, and plasticity is bad here, so it's got more of it. But when we looked at this distance from the optimum, we find that the temporal regime does better, significantly so in the cadmium diet, than the spatial regime. So temporal appears to have more bad plasticity, but it also appears to be better adapted. So how can both those things be true? Well, one way that can be true is diagrammed here. So imagine that the optimal level of expression in both diets was quite low, and so ideally you'd have no plasticity and have low level expression. But initially, when you enter these regimes, you have a reasonably high level of expression. Selection is driving down expression in both diets. But if that occurs faster in one environment than in the other environment, then you get improvement in both environments, but are left with this signature of apparently bad plasticity. Okay. So to conclude, I showed you that evolved differences between populations that live on only one of these two diets, that the evolved differences between them seem to be counter to the plastic responses of the naive population. I've shown you evidence that there's adaptive evolution of expression, that those heterogeneous regimes appear to be closer to the optimum than the non-adapted, homogeneously selected regime. And I've shown you some evidence of adaptive evolution in plasticity and expression. That sometimes happens, but it doesn't always happen. And I've shown you that there's at least some hint that plasticity may be evolving differently with spatial heterogeneity than with temporal heterogeneity. In particular, with spatial heterogeneity, it looked like there was more evidence of adaptive evolution for genes where we expected increases in plasticity. But when we looked at genes where we expected reductions in plasticity, it looked like there was more adaptive evolution occurring in the temporal regime than in the spatial regime, but that actually didn't translate into the naively expected pattern on plasticity. So with that, I'll end by again acknowledging Yuhan Wang, who did all this work, and people who helped us think about the project and funding, and I'm happy to take any questions.
Say more about how you came up with a list of candidates. You mentioned these candidates.
Yes. Well, so for what are the candidates, or how did we find the candidates?
Both, right.
So first, how we found the candidates. So when we're looking for genes that we expected adaptive plasticity, we look for cases where in cadmium, say, expression was low, the diet-specific answer that's adapted to cadmium has a low level, and the diet-specific answer that's adapted to salt has a high level of expression. So we think that if selection is doing just what it wants, and it's not constrained to worry about the other environment, it creates a difference. So it seems like ideally what you would do is have plasticity to respond that way. And we use a similar kind of logic for genes where we expect adaptive reductions in plasticity, where both diet-specific ancestors tested in their own environment, for example, have low expression, but when you put them in the wrong environment, they both show a high expression.
Yeah. Can I follow up on that question? I'm interested in the function of some of these genes. I'm guessing most of the metabolism. Did you see any variance with respect to the overall function?
Yeah, so we did the thing that everybody does, which is you do a GO thing, and it's all gobbledygook to me, to be honest. So I can't. There's nothing sort of like stupidly obvious that comes out. I think someone who knows something about this stuff might be able to interpret it. Unfortunately, that's not me. Thanks for listening.
Thank you for